Hello, everybody. So in this one, we're going to talk about the differences between an antibody and an antigen. I think the important thing to remember about antigens is that they are not you. We use this term non-self. Most of the time, they're surface proteins on the surface of a bacterium or a virus. So if we were going to draw maybe a little bacterium here, this little green thing could be the protein on the surface of this bacterium. Um, and the result is an immune response. Your body will recognize these things as not being you, and it will start producing something called antibodies. Antibodies are made by a type of cell called a lymphocyte. These are cells found in your lymph nodes. And specifically, the type of lymphocyte is called a B cell. So these B cells will make these antibodies. They are, as we mentioned, are similar to antigens. They're also proteins, and they're globular. They're made of 1, 2, 3, 4 chains of polypeptides, and at the end of them, there's a binding site. This binding site is specific for each type of antigen. So every time you're infected, you make a different antibody for that infection. So if you remember way back when we were talking about transcription, translation, and protein synthesis, there's a section of DNA called a gene that eventually will code during the process of transcription for a messenger RNA. This will eventually leave the nucleus to a ribosome where that protein, in this case, the antibody, will be made. Now, it might be hard to believe that you have that many genes for all the possible infections and all the possible invaders that can attack you. So what your body does that's pretty tricky, if you remember, is whoops, I'm not sure what happened. Right. So if you remember, we can take out part of the messenger RNA and then we can glue it back together. We can stick them back together, which will create a different message, right? This will be a shorter amino acid for sure, or a shorter polypeptide for sure, and it will be a different chain of amino acids in the end. So that's how you make so many. Um, and the way antibodies work, if I'm going to draw, let's draw our bacterium here. These little circles represent um, the surface proteins, so the antigens. So here's the binding site to that antibody, like that. So maybe one's attached there. Maybe there's another one attached here. Maybe there's another one attached here. And when you have a whole bunch of antibodies attached to the outside of you, there's not a whole lot that bacteria can do. So it's going to make it inactive. It will also stick it to other bacteria. So it's easier for those macrophages, right? Those phagocytic leukocytes to consume it. Also, if you can think of it, they're like little flags on the end here to help identify, hey, listen, look what we found. This is not supposed to be here. So that is the difference between antigens and antibodies.